Being a scientist in a Johnson and Johnson, and we have been involved in the vaccine research for a very long time. So we believe that vaccine is the one of the most important and the potent solution that we can have. But are we going to have it very soon, or is it going to take a long time? That's the debatable question, and that's $1 million question actually. And everybody is trying to go for it, and let's see how far we can go. And I want to take you along the journey, like how it has been developed, what are the difficulties, and then what are the new approaches that the vaccine research has come up. And with that, I would like to share my screen. So I don't want to go into like a minute detail of COVID-19, that like the what the disease is. I will go briefly over it and what are the different approaches of vaccine that going around the different world. And I'll be focusing on two main approaches, and that is very promising in the current situation. And one is like developed by Johnson and Johnson, and another is called a company called Moderna, which is situated in Boston. And these two type of vaccine, how they are doing, what is the molecular basis for it, I will try to give some light on it. Okay, now let's start. So like if you really this is one of the most important invention that people are looking for it. And like it is one of the most not only important, it will be very critical in saving the world population right now. Until now, there has been vaccine which is like it takes a very long time to create, almost like more than 5 years. So we are racing against time, and we are trying to get this developed within a very, very short time. That is like almost like 14 months, 9 to 14 months. So that is very, very important, that otherwise we will not be able to like save the many population around the world. So this has been a very biggest, one of the most speediest race in the search of any kind of medicine in the world right now. And we have the technology, and everybody is trying to leverage the technology, but still we are not sure whether it will be successful or not. And if we fail, then there will be like a big loss in the population wise. So this is one of the most urgent and will require tremendous amount of collaboration. And when you talk about collaboration, it is collaboration across the world. It's not about one company or one research institute is doing. They will not be able to successfully do it. If they are successful also, they will not be able to successful in the sense of manufacturing it. So we need to have collaboration, and it has to be across the world. And that is one of the very critical aspect that people have come to know with this pandemic. And then the most important question is this, like we need to supply to the next 7 billion people, 7 billion people across the world. So maintaining and starts, nobody like many people are not having that capacity. Okay, so that will be optimistic, because we cannot be scared at this moment. We cannot be pessimistic. And so that in the next 9 to 40 months, we come up with a solution which can be shared. Like I believe in that. And that's what I want to share with you. And what exactly is a coronavirus? I would like to give you some brief background, because otherwise picture. So what is a coronavirus, and how does this outbreak compare to the SARS outbreak in the 2003? So there was an outbreak of severe acute syndrome of like a acute syndrome. Okay. So that where like almost like 8,000 people has been killed. Uh, with the COVID-19 situation. Okay. So that situation was something very new for a new generation in 100 years. It hasn't happened like before. At the COVID-19, it has crossed millions. And although the casualty compared to like SARS is more, but if you look at the population that has been infected, then the ratio is much less. That is one of the very big positive thing. Then we can take it out from this pandemic situation. And I will tell you why later on. Okay. And we know that the Wuhan in the Hubei province in China is the origin of it. Okay. and that where everybody knows it, and it is a virus which infect human. And there are a lot of political as well as political questions are there, whether it has been come out naturally or it has been planted. That thing is a very controversial thing that I don't want to talk about that. But it's like we know that it has originated from the Wuhan, and within a span of like 2-3 months, it has came across the Atlantic to US, and Africa, as well as like a Europe and so whole Southeast Asia. But that means that it is spreading without any barrier of temperature, without any climatic conditions and all. So people were expecting that maybe like climate is going to stop, but it was not stopping it. Maybe the humidity is going to stop, but it is not stopping it. So this is something that it is completely new in a way, and the people are not aware of it, and it is showing a different kind of characteristics in the sense of physical characteristics as well as like infection rate. It is not like confined to any particular area or the region. It is just globally it is infecting. So that is a very, very scary thing. But at the same time, we have found in the Janssen and Janssen, like once it was detected in Wuhan, then we came to know about the sequence was released by like a January, and the beginning of this year, the sequence was released by the Chinese authority, and that sequence has been shared throughout the world. And it is, we know that it's a mRNA virus, and the mRNA sequence has been shared throughout the world. All the scientific organization, they got the sequence of it and they analyzed it. And, and in the process, they have analyzed the sequence of all the different variants of COVID-19 across the world. Okay. And the one of the very important thing they found was the mutation rate is very minimal till now. People expected that the COVID-19 which has been infected in India and the COVID-19 virus which is infected in America will be completely different. But it was not so. It is very minimal differences there. And the virus, if you know about the virus is the only living thing, you can say, which has survived throughout the a civilization, not only throughout the living civilization of the whole world. Every species got like extinct. There are many species which was been there, and it has gone extinct with time, and human beings are the new one, and it will also go extinct. But the viruses, they haven't been extinct till now. Why? Because they have the capacity to mutate themselves and adjust to the situation, adjust to the climate. Okay. And because of that, getting a solution against a viral disease is a very, very big problem from a medical point of view, from a medicine point of view, because they mutated. So once you have a molecule which will be targeting that virus, for example, then if it mutates, then it's not going to work. So that's why it's very, very difficult thing. And the Janssen and Janssen in Netherlands, the headquarter of Janssen, where the vaccine research happened, they have found that the mutation rate is very minimal because this virus has a skill of editing itself. If there is a mutation during the replication happens, they edit themselves, they proofread itself, and they get back to the original one. So that's a good news for us, because now we can, if we design certain vaccine, then it will be completely safe in a sense of they will not change. So the vaccine epitope will not, we don't have to change the epitope many times or developing many, many different candidates for it. So we can have only one candidate which would be universal character of that virus, and then we can go ahead. So that's a good thing for us. At this stage, the outbreak most associated with the novel coronavirus has been the older people, okay, and many of whom has a pre-existing one. So that also reflect one thing, that the virulence of this a virus is not that much. If you're healthy enough, then it cannot destroy you completely. Okay, but if you have some precondition, then it can go over it. That is a fact, and that has been proved across the world, because the victim, most of the death population, these are elderly population, as well as who has some precondition, and because of which their immune response has been compromised. Okay, and the next, how are these people are going to be treated, and how can the other best protect themselves? So the now question is that can we protect these people? Can we protect ourselves also? What will be the solution for it? Okay, that is the very big question that we are trying to find for it. So recently, like at the WHO recommended that maintaining the basic hand and respiratory hygiene. These are the two things that people has been, the WHO has told everybody to maintain it. And safe food practice, that is everybody know it, and the avoiding the close contact, and that is the social distancing, and that people have been talking about. This social distancing is nothing but like it has been practiced in during the SARS pandemic in the North Africa, and they have successfully came out that social distancing really help, and that is this not about, it's not a new it's not about nonsense. It's like it's very important for saving the humanity at that side at this time, and this is the only way, like people have come to know and people have concluded that this is the only way that people can be safe in the absence of any kind of vaccine or any kind of antiviral drugs. So we should not be like we should not overlook the social distancing, that aspect. And I know that in India it is not that much uh, followed by people, and it is definitely a political question as well as economical question also. So any country cannot just survive in social distancing, or if the economy is going to be hard, and once the economy is hard, then it's going to affect the whole country as such. So it's very important that we should maintain, we should drive the engine of economy also, but at the same time we need to maintain the social distancing, so that we are not many people are affected, and then until the vaccine is there. So this is very, very important, and we should not be like uh, we should not overlook this thing. And I want you guys also to follow in India, as well as we are here in the US. We are, everybody is working from home, but next month onwards I have to go to the office. I will go in between, but everybody over there is also having a distance of at least 6 feet. We are not allowed like 4 or 5 people in an office, in a room. We are not allowed like to talk without a gloves, without a mask. So those kind of thing has been strictly followed over here. Although there are lots of uh, death occurred in US because initial response was delayed, and that those people who have died also, they have the precondition. That is one of the reason also. But now they are maintaining it very strictly, and I hope that everywhere in not only in India, across the world, they maintain it, because we have to do it. Otherwise it will be very, very difficult to contain this one. And now the solution, is the vaccine a solution? Is it the only solution that that we have? In my opinion, that is the only solution, because antiviral also, yes, many people are working on that antiviral aspect of it, and there are many other aspects people are working on it, but I don't know how far it will work at that sense. But since I'm involved in vaccine, I believe that the vaccine is the only solution that we have, and it's going to take some time. And I just want to tell you in between, like, if you have any question, just let me know. You can interrupt me and you can talk to me. Okay. So otherwise, I was thinking of making it a little bit more interactive. So if you think of, or there is, if you think of like uh, anything, then let me know. Okay.
Yes, definitely.
Okay. Thank you. So the treatment saves lives, but the vaccine saves population. Okay, so it's like if we have a vaccine, it's not about you. It's you are saving the other person also. So in that way, we are saving the population. So it's not only about individual safety; it's about the saving the whole population, saving the whole human race in a way. So what you get a vaccine, when you get a vaccine, it's not about yourself. It helps ensure that the transmission chain stops with you, and that helps keeps your loved one safe and healthy. So the transmission, which is, if you are infected, so once you are infected, virus is going to replicate in your body. Okay, and once it is replicated, it will try to go out of your body, And that thing, if you can stop that rate of application, rate of infection, if you try to stop that, and that can only be possible with the vaccine. And if you stop that, then you are not infecting the next person, and that way you are critical thing now that we need to follow. And that's why the social distancing is very, very important. It has been developed, and there are many across the world it is happening. Okay, so what are the different what are the people has been making the vaccine till now? Okay, so one, the whole inactivation is happened because of one virus. How it is done? So that virus is completely inactivated. That means it is, in a way, it is killed, and then it is injection that the killed vaccine is going to enter your bloodstream. And the bloodstream, what will happen that the humoral immune response will be a T cell to kill it. Okay, and that's the way to stop in any kind of like a vaccine treatment. And another is a attenuated. Attenuated is the example is measles. Attenuated is like they don't kill the complete, they don't kill the virus completely. They killed it enough so that not infect your body. They cannot make the disease happen in your body. They cannot they will not make harsh kind of effect to your body. So they kill it inside your body, and once that is inside it, and that the same way the immune response will be activated, and the antibody will be raised against that live attenuated virus, and then it will be saved from any kind of infection from the future also. Because once the antibody is formed, then what happens is that we have a different kind of cell called memory cells. The memory cells are the cells which will always remember that antigen, that is a live attenuated virus. And whenever you have another infection, for example 2 years, 3 years, or 10 years later, then what will happen, that will again the antibodies will be raised against it, because you already have the memory. So you have the antibodies like an army kind of, all the soldiers are there, they will come and then kill that virus, so you will not have that infection again. So in fact, the vaccine, once you have the vaccine, you become protected throughout your life. That is the thing that people say. And at least for 10-20 years you really get safe from that kind of disease. And sometime later on, after that 10-12 years, you need to get another booster dose, because you need to have that kind of dose to elicit your immune response. But generally it stays throughout your lifespan. So that's why the vaccine, once it is happened, once you inject with vaccine, it is safely said that you are protected for a lifelong. But there are many viruses which get changes every now and then, so you have to take it like every year. For example, in US, the flu vaccine you have to take every year, because the flu changes very much and the virus changes the epitope. So once you have a one vaccine today, that cannot be effective in the next year. So many people take vaccines, some people don't. And in fact, I don't take. Some people take it. Initially I used to take it, but now I don't take. It's like sometimes it happens, like you don't necessarily need to be vaccinated every year. But people take it. That's the general rule for any kind of flu vaccine. So the next is these are the most classical. Oh, another classical way is the subunit, the hepatitis. For example, in hepatitis, the virus, you take one part of the virus and that is injected inside the body, and that will elicit the immune response, and the antibodies will be created, and you are protected. So these are the classical way which has been happening since the happening of like a polio virus for the last maybe you can say like almost like 100 years. Okay, so people have been developing the vaccine on this basis. So now there are different different approaches are there. And who, because of the COVID pandemic, today they have last week they have declared that there are like 150 50 COVID vaccine initiative is going around the world. 150 initiative, different way people are trying. Okay, but we cannot just go over every different way, and I would like to focus mainly on the very modern and the recent way that people have been going. Okay, so okay, so before going into the new technology, I just want to talk about the how the vaccines are developed actually. So as I told you, vaccine is like either they are live or a attenuated or a partly part of the vaccine is injected. So that part, or that whole vaccine, the full attenuated one also, you need to culture it. You need to go for a designing the molecule. Okay, you cannot just put anything into the body, right? So you have to design in a such a way that it is not going to harm you. And the second question is that once you design it, how you're going to make it. And these all cells, all the virus has to be produced in a such a way that it can be produced in a larger scale, so it can be served to the millions of people. It's not about one or two people, it's like a millions of people has to be done. So most important thing is that once you design, then you do some early development is the sense of that is the research that you do, so that you can have a process which will be scaled up in the sense of thousands of liters you can manufacture it. So all those during that early phase development, you try to identify different critical parameters. Okay, so those technology has to be transferred to a larger scale. For example, you all understand, when you, for example, you're cooking a chicken in your home. Okay, you're cooking it for 3 4 people, 100 people. Okay, so 100 people making the chicken for the 100 people, you need to have that. So only few people can do it, only the cook who are in the restaurant, they can do it. But when you're cooking regularly at your home, the cook know what are the process, what are the parameters. That means parameters in the sense of cooking will be like how much spoon of salt, how much oil, how many chicken, and one spoon. For 100 people, you have to add maybe like 10 12 or 15 spoon of salt. So that thing, how you're going to know that thing, how that, okay. And there, those process parameters are transferred, that is called technology transfer, to a larger unit, which will 3,000 liters of all the culture of the live or attenuated vaccine. And that process, after culturing it, after that, they have to manufacture it in such a way, the physical property, the chemical property of the vaccine remains same. Okay, it cannot change. Once you, if the process, or if the vaccine which has been produced in a larger scale, it changes the chemically, or like a physical chemical properties are changed, then what will happen? It comes in your body, because it will recognize, it will be like a more virulent, or it will develop in a different way. So you need to maintain a standard in such a way that the properties of that virus, of the killed or attenuated or a part of it, doesn't change. So that is a very critical phenomena once you discover the molecule, and that is called the process scale up. And once the process scale up is done, that is the phase 1 campaign, where you try to see the immune response of the people once the vaccine is done. Okay, so that you will give us very small population. You will have a sick population and not so sick population, maybe like 50 or 100 people, and then you will go for thousands of people, then you will check it out. And all these, like up to phase 3, they will check in a different kind of population, and then once you get the proper result that it is really affecting, and then the FDA is the regulating body of US, they will check everything, and they will say that, okay, you can go for manufacturing and you can sell the product. That also, it's like a long process. The whole process actually takes around almost like more than 5 to like 8 years. Sometimes it takes 10 years also. And now in the COVID situation, we are racing it like almost like 9 to 14 months we are trying to do it. So whether this kind of development will be possible, whether the whole life cell kind of approach is possible, I don't think it will be possible. Okay, but there are many people who are trying it. Whether the attenuated will be possible, that also I don't think it will be possible, because like a whole scale up thing, as well as like a making the from the part, the classical way of making, and the checking with the people itself, is going to take almost like 2 years to go into a first clinical trial. So that is not possible at all to maintain the race. Okay, so we need to have a different approach for it. So and then generally, like once you have the process done, and you have transferred in the like a manufacturing unit, so how the viruses are purified. So most important thing is that generally all these viruses will be cultured in a cell line which will be compatible with the human cell line. Okay, because they will produce in such a way that the virus is not modified. Okay, this is very similar to human cell line. So once those cell lines it produces, it is very important that it is produced in a cultured way. In the culture it will be produced, and once that produced, we need to filter it. Filter, that is clarification filtration by depth filter. Okay, depth filtration, I don't know whether you know or not, but that is one of the very, very important thing in like any pharmaceutical industry. And like you have to filter the product that you have culture, and that after filtration then you have to do like there are a lot, it's called ultrafiltration and diafiltration. That way you can concentrate it, because you'll be culturing like thousands of liter. You cannot put thousands of liter in a column because this is like a small molecule. So you need to put it into concentrated form. Then you have to concentrate the volume. Then you have to pass it through different columns, which will bind to that protein, and the rest of the things are not going to bind. Once that is bind, then you take it out and then you do the DNA-ization. DNA-ization means you have to, first of all, the cell has to be lysed in a way that the cell will die and the protein will come out. That digestion has to be there, and that digestion will kill all of them. And then after that you again fill it up and then filter, and then you go through different kind of process, and then ultimately you have the vaccine which will have only that small molecule. Okay, so all this process takes almost like more than 5 to 6 years if you're very lucky, if your all the clinical trials are like a positive. For example, you are checking 100 people, okay, and then like you are getting like almost like 80% positive response you are getting, okay, then it will be considered as a okay successful campaign. But if you are getting only 20% like successful and rest of the 80% population are like affected in a very different way, very different in the sense of like they are having they are having some kind of fever and all, it's not acting positively, then that campaign will be stopped. Okay, then you have to go for another one. So getting a success in a vaccine research, not only vaccine, in the nowadays all the next generation drug, it takes very long time. It's not very easy to get it the like a positive result within a very short time. Okay, so at this situation we know that we have the classical pathway, I'm telling, classical approaches, which will take very long time, and it is like it will be very difficult to assess the situation without spending that much of time. Okay, so but we don't have the time right now, we don't have a bigger, we are racing against, people are dying, so we need to go for a different approach. Okay, so what can be the different approach? So already the WHO has told that it's almost like 150 different approaches across the world. Okay, there are a lot of people are trying to do it in different way, but like are on 150 approaches will be successful? If it is successful, then it is definitely good, but the chances of success, will it be dependent on like it will all depend on the efficacy of the product, right? So you cannot just say that 150 are going, so we will be lucky within next 1 year we will be getting it, but chances will not be like that. Then not only the efficacy, then whether then another question will be, are you going to able to manufacture it in such a scale that you can supply the vaccine to the rest of the 7 billion people? So are all those 150 initiative have that capacity? Is it possible really? Maybe no, or maybe yes, we don't know. So what are the different the new vaccine, that new way of vaccine that we are trying to do it? Okay, for the last 100 years we have like all those classical pathways, all those like attenuated, killed, and all those stuff, the whole virus has been used. But now the recently for the 30 years there are different approaches. These are like a DNA based, RNA based, and a vector based, that is advec and a PER, like a cell line, that is a Janssen a proprietary, the third one. And the RNA is the different one, that is the Moderna company is doing on. And the DNA, many people are working on the DNA vaccine. And if you look at the traditional approach, it's a very long, it's very complex, and it's a costly development and a production. So if you look at any kind of vaccine approach or any kind of medicine, ultimately it goes down to money. Okay, so yes, no doubt you are trying to save the population, but like how much money you're going to spend and how much money you are going to get out of it. That's the main thing. That's the economics. Okay, that it plays everywhere, every phase of like a medical world. Okay, so first thing is that in the traditional way it is slower, But in the nucleic acid, the DNA or RNA based or any kind of vector, it is much faster compared to like a slow compared to the traditional one. And as I told you, that in the traditional way, we need to customize it for each vaccine, like in the sense of, because most of the time you are trying to you are trying to give a attenuated or a killed or a part of it, right? And the virus changes, virus mutate. Okay, so you have to customize that every time. All the flu vaccine every year the US population take is manufactured in a different way, because the flu vaccine, the flu virus which is affecting now, the next year it will be completely different because they also mutate. Okay, although the process may be same, but it has to be designed in a different, it has to be customized for the recent one. But if you have a DNA or RNA one, you don't need to do it, because the DNA RNA, because you are going to take only for a specific part of it, so that is not going to change. Okay, and the scalability is very important when you think about manufacturing it for a larger scale. Okay, if you're thinking about like giving it to the millions of people, then you need to have a process which has to be scalable in the larger quality, a larger like a quantity. But in a vaccine, for this one traditional vaccine, you need to have very large like a facility. But in the DNA or RNA, you don't need that much of big facility like compared to the traditional one. And the cost is also like it's like almost like 200 to 1 billion dollars you need to have for a traditional vaccine to be developed. But in case of RNA and DNA, the cost is also minimal compared to the a traditional one. And as you know, the high volume will be much more important in the traditional one, but in the RNA and DNA it will be much less. So, and another thing is that a humoral response many requires adjuvants. Okay, in the in the in the traditional vaccine, most of the time it is the B cells, like antibodies, are activated. So antibody will has to be formed. Antibodies are the humoral response of immune response. But if you talk about the DNA RNA, most of the time what people have found, it is not only ignited or not only like elevated the immune response through antibody formation, but it has also found that the T cells are also elevated. Okay, so T cells, when T cells are activated, so that it happens is that they're like a two-prong attack. It's that antibodies are also attacking and the T cells are also attacking. So you have a much more better response, better immune response compared to the other one. Okay, now the question is, uh, now yes, we have a two different we have like many different ways of making like a vaccine. But in the recent COVID pandemic, I have selected like two main approaches that people are following, people are trying to do, and people are expecting that it will be successful. One is from the Moderna, that is a company in Boston. They are based with the mRNA approach that I described you before. And another is the Johnson and Johnson approach, that is a vector-based advect and a Parco cell. That's the different cell line that is used. And these are the two approaches that I will try to focus and I will try to explain with you. And these are the most modern, which has approaches that is happening in the vaccine world right now. Okay, so in the mRNA platform, what happened is that, as I told you, the DNA is what is the operating system? Operating system is the system which will guide the principle of designing the whole thing, right? So we have the DNA, and DNA, we know that it stores the information, and that information, it is translated into a mRNA. mRNA is what? We are trying to say that it is a software. So mRNA is temporarily set of the instruction for the cell to make protein, and mRNA is made using the DNA. So DNA is act like a storage. You have the CPU, and the software is the one which is running, and the proteins are what? These are the applications. If you really think about the operating system, this is where it goes. Okay, so in the DNA, mRNA, and the protein, and the whole thing is called protein synthesis, and this is the basis of everything of all the life. Okay, so this principle has been exploited in the RNA platform. So what are the advantages of it? So most important advantage is that the mRNA encodes every protein in the proteome, right? So all the proteins that you have or the virus has to be encoded by the RNA. So if we have the RNA, we know what protein it's going to make. So another advantage is that this process is a completely natural process for all the living creature, right? So everybody is going through protein translation, protein transcription and translation. That is the protein synthesis machinery is there in your body. It's a very natural thing. There is nothing artificial about it. So the body is going to accept it in a very easily. It's not that you are forcing the body to do something else. It is something that you are asking the body to do, which is very natural to them. So it becomes an advantage to use that kind of approaches. Okay, and the simple chemical makeup, the makeup of the whole RNA is also very simple. When you design the RNA sequence, you are designing in a such a way that first of all, it will be recognized by the body. And the second thing is that it is not going to initiate any kind of immune response, that is which you don't want it, huh? And the other aspect is that you are trying to make it more stable so that the body immediately recognize and try to translate it into a protein. So that making it stable, they try to prime the most of the time the N terminal is capped with some kind of uh, amino acid. I forgot what amino acid they use it, what codon they use it for, make it stable. But in the generally the prime N prime N, they try to modify it in a such a way that it is a little bit more stable. And the second thing is like what is the pharmacologic feature of it? So is it potent enough? And the dose dependency is also there, and is it like reproducible, and are they expressing the protein which we wanted? So most important thing is like once you inject the RNA, whether it is potent enough in a sense of developing the immune response that you want. So how it is going to do? So once the mRNA is inserted, that will be recognized, and the protein synthesis machinery is going to work on it in such a way that it will translate into a protein. So that protein, is it enough in itself? Enough in the sense to elicit the antibody reactions? So that will define the efficacy of it. So that means the dose of mRNA that you are injecting, that will decide how much response you are going to get it. So dose dependency is there. Okay. So, and then the question of reproducibility. So once you design one mRNA, for example, you make a certain sequence of mRNA, and then you know that it's going to act on it. So you saw some positive result also. Now the question is it really, will it be able to reproduce in the same way in the next time? Yes, the question is yes, because the designing of the mRNA, you can do it in a computer. You can do it in silico, you can design it. So that in silico design is very, very consistent rather than designing in another way, right? So it is reproducible. And the second thing is whether they are expressing the right protein that you are expecting. So in this case, they are trying to express the spike protein of the COVID-19. Okay, that is, they have different spike. So Janssen is also working on a different spike protein. So these are all like something that I cannot talk about directly. You can find it out maybe, but they will not. It's like a proprietary kind of thing. So what protein they are trying to clone it, that I cannot disclose it, but they have different protein. So whether that protein has been expressed properly in a way that antibodies are raised against it, that is the next step. So all these criteria, for example, has to go for a traditional vaccine, also go through this kind of procedure. But the consistency as well as the reproducibility in the mRNA way of approach, it's much, much higher. Okay, because in the traditional way, it may not be one batch may not be the same as the second best. There is a chances of error or the change or the chances of like a making it different is always there. But with the RNA, since you are making it, designing it in silico, so chances of error is like negligible. Okay, and then the if you check that, if you try to find out that 20 nanogram or 25 nanogram is not affecting or not giving you that much of response, then you can increase it like a 50 or 100, and that cost of like a 50 nanogram to like a 25 nanogram is not that significant. Okay, and then you can manufacturing in a single unit platform in the sense of your whole process is same, and you have a platform process for all the RNA molecule they are testing, so you don't have variability. Okay, so that also reduce the cost. Okay, so ultimately the cost of production become much less compared to the traditional one, and the time of doing also become much easier, because you can check in silico itself, you can design millions of a candidate, right? You can mutate it, you can change the residue, you can do all sort of different combination, you can do it, and then check which is much more potent. Okay, so within 1 month, 1 month you can select, or you can go through a screen of like thousands of RNA molecule. Okay, and whatever you get better, that you go for a manufacturing. So you, but if you try to go for a traditional one to check for like a 1,000 molecule, to screen for 1,000 molecule, it becomes a huge task. It will take the 2 years to do it, because you have to produce each and every one in a different way. Okay, so that is one of the most advantage for the nucleic acid based like a vaccine approach, and that's what the people are expecting, that it will be, there is a chance of successful for the COVID-19 also. So from the idea to the drug candidate nomination, that is what I was trying to tell you, that they got screening a candidate, it's a very, the span of time they have to spend is very less. So once the like we came to know about COVID-19 by January, right? And the sequence has been done, like a sequence has been published by end of January. I think by that time, by February itself, they are able to design, the drug candidate which they will go for a clinical trial. Within 1 month they were able to do it. Okay, that is the fastest way of doing it. You cannot just have it at classical way of vaccine development within that sort of time. It's very difficult. So the idea, the full control over mRNA design, and the experimental velocity, that is the like a how fast you can experiment it, and the massively parallel hypothesis testing, that is the idea that you are trying to test. You can test it in a parallel, different idea you can test it in a parallel, and then you can choose your drug candidate. So the whole lifespan of the idea to the drug candidate designing is like minimal compared to the any classical way. So that's why the people are expecting more out of the Moderna research. And if you know, it is one of the Bill Gates has funded this heavily on this Moderna, and that he's expecting that it's going to be successful. But whether it will be or not, that we don't know. And the second thing is that early mRNA drug development. Once the drug candidate is nominated, for example, you have successfully designed a candidate, and you know that it is going to be efficacy is going to be good, and you can manufacture in the larger scale also. Then you need to go for a clinical trial, and once you for the clinical trial, you need to have a huge amount, right? So you first of all, you have to have a continued influx of mRNA development candidate, because in the sense of if it fails, then you need to have different candidate. You can have that parallel kind of designing as well as testing, and then move the DC through a good laboratory practice, that is GLP toxicology study. So toxicology study is the one of the initial studies that that the pharmaceutical industry do, is that they try to inject it into like a chimpanzee, that is like a more like a human, and they try to see how much bad effect or like an immune reaction which is not desirable is happening. Okay, so they try to see how much bad effect is there. Okay, in a animal study. Then they conduct a human proof of concept study. That means they will try to inject into a human in a small population. Okay, not very huge. And Moderna has that, they have declared that they have done it up to 45 candidate, I think they have done it till now. They have injected the first phase 1 clinical trial, and that clinical trial, after like injecting, they will check it, like they will check it in the, they will check 2-3 parameters. One parameter is that the doses are enough, whether the for example, whether it's 25 milligram or microgram or like 100 microgram is affecting. So those will be, doses will be selected in a way, 2 doses will be injected in 2 different population, and then they will check how the immune response is happening. They will try to check with the population which doesn't have any disease, and they will also try to check with the population which has the disease. Okay, so this way they will compare it. So, and then you will have the first proof of concept in the sense of the candidate is okay, candidate is good or not. Okay, so like then the quality will come, they will check it for different oh, I'm already, it's too long or what? It's
No,
then
no,
the
fine,
is it
fine.
okay? Then they,
It's fine, sir.
okay, okay. So the quality will be done, and then they will check with the scalability. Scalability is that whether they can produce in the larger scale, and the speed. Now the essence of COVID-19 vaccine is the speed, right? And the speed, we have to produce it within a speed like a limited number of time, and then the cost. Cost is definitely less compared to the traditional vaccine. So, so these the key milestone that the Moderna to the advanced mRNA, that is 1273. 1273 is the spike protein that they are working on it. So like a first milestone is like on March itself, the NIH announced the first participant in the phase 1 study. Uh, and the 6th April, the phase 1 study of this completed, and the 18 May, the last week, they announced that there are some positive result from the phase 1 mRNA studies. Okay, so there are some controversy going on, but I don't want to go into that. But they are the first one who showed some positive result with the mRNA way of vaccine. Okay, so we are expecting that they will be successful in the next clinical trial, and then maybe the beginning of next year we will have some positive result as well as some vaccine too. And the next I want to go is the J&J initiative, the Johnson and Johnson. That is a completely different way of making the virus. So in the J&J, what they are using is the platform that they are using has already been tried with the Zika virus, RSV, and HIV. Okay, till now, in fact, they have already developed a vaccine with the Zika virus and the RSV and the HIV also. They are trying to do it, and that platform has leveraged to design a new technology, a new vaccine for the COVID-19. Okay, and this COVID-19 vaccine the whole platform for the Johnson is known as ADVAC and PAR-C6 technology. Okay, and what is that? That I will tell you now. So ADVAC PAR-C6 technology is like ADVAC is for the adenovirus. Adenovirus is the virus which causes common cold. Okay, and they are very, very good in transporting into the humans. They infect the humans. So they naturally infect it, and they causes the common cold that we have it. But the Johnson and Johnson, what they have done it, they have they have genetically engineered the adenovirus in such a way that it does not replicate in the humans, and nor it causes diseases. Okay, so but it has the capability of infecting the human cell. It will go inside the cell, but it will not like cause any disease, nor it will have the replication capability. So this technology, for example, I just have a small video. So this vector and the genetic code is over here, and then what is the technology is like inside. Once the adenovirus is modified, genetically engineered, which doesn't affect, in fact, which doesn't have any a capability to replicate, that adenovirus is taken, and inside that a vector is inserted, which acts like a vector, and there the RNA sequence of the spike protein has been inserted. That is the genetic code for the COVID-19 spike protein has been inserted into the adenovirus vector, and which once it's infect the cells or the human cells, what is injected, that RNA will be translated into a spike protein or any other kind of protein. For other viruses, it's different, but for COVID-19, it's a spike protein. And once that is translated, and it will be recognized by the antibodies, and the antibody will be formed against it. Okay, it is same like RNA virus, where the RNA has been inserted, but here the RNA has been inserted inside the vector, and that vector is nothing but an adenovirus. So the adenovirus, since it has the capacity to infect cells, so it has become much easier mode of transporting inside the cells, inside the human bodies. Okay, so that is the biggest advantage for this one, and it has already been tested in some other vaccines. So we are leveraging on that. So now the step 2 is, what is the step 2? So the T cells are also elevated, antibodies are also formed, and these are the antigens that is translated. Okay, and then the antibody as well as T cell is going to act on it, and the immune response will be elevated. Okay, so most important thing is that it produces the B cells, antibodies, as well as the memory B cells are also, as well as the T helper cell, cytotoxic T cells also. That means it is elevating, it is igniting the whole immune response in a way. Okay, it's not only dependent upon only the antibodies. Okay, so it is activating the T cells, and not only the T cells, T helper cells and the cytotoxic T cells, because they are the one that is going to kill it. And the memory cells are also there. So it is elevating the whole immune response. So we are expecting that it's going to last it for a longer time, and it will have a much more safer response against the COVID-19. Now, the next thing is like a step 3 is, in future, what will happen is that this is the antigens, okay, and that is like a virulent thing, and the antibodies are coming, and to be, this other cells are also, T cells are also coming, and it will have a memory cells, that the memory cell are going to recognize this one. So whenever there will be a other like a real infection is there, the response will be much faster and more effective. So like we are having something that it is very much natural to our body, and it is easy to transfer inside the body because it is adenovirus. Because adenovirus has the capacity to go inside the human cells. And the second is that the response is much faster and more effective to fight infection and preventing the disease. So, what is the PAR cells? So these all vectors translated, it's transformed into a PAR cell line, and PAR cell line is the one which will make millions of vectors, which contains the epitope, the sequence, and generally all the cells which we have it, which we try to culture in the pharmaceutical industry, is very different. They will culture in a solution. Okay, so it will be a floating suspension. It doesn't need a surface. To most of the time the mammalian cell grow on a surface. Okay, so you need to culture it in a plate, but these has been modified in a such a way. I think Janssen & Janssen only has these cells which can be cultured in a suspension at a high density. Lots of millions of cells can be produced. Once you have millions of cells, that means you have a millions of vectors. That means the cost of production become much less, in a way that you have millions and you have to give it to millions and all. You don't have to culture it like too many times to supply it to the millions. And vectors like AdVec can replicate at high numbers and concentration in persist cell line, and providing the ability to rapidly upscale the production. So that is the main crush of, or the main advantage of Johnson & Johnson, is that they can culture in a larger amount in a very short span of time, and they can get millions of doses out of it.